we'll hope for better luck with this time. So I'm going to start with a quote which I think it seemed appropriate in the context of this talk. So I spent a lot of time while preparing this talk looking through a lot of old papers, like dating back to the 60s and onwards, and basically seeing that all of the same problems that we're talking about with multicrystal and data processing and data collections that we're facing the same problems that they've already had to deal with many years ago. So I mean, I could have spent the whole talk giving you going through like some of the old papers, because that would be really interesting. But I've narrowed it down to this one specific example because it demonstrates a lot of the areas that we're still interested in now. So from the three-dimensional structure of foot-and-mouth disease, I think it was published in 1989. I think it was collected at Daresbury. So they comment on how they had problems with the particularly small size of the crystals, at least by the standards of the day, led to very weak diffraction. And if you think back to Graham's talk, you can see why that is. And also because they had large unit cells and so on. The crystals were particularly radiation sensitive, so they had to be careful of radiation damage, and so they were only able to take a single small oscillation shot per crystal. So they had to, in total, they had to examine over 500 crystals, of which they, I think, eventually were able to process 106, contributing to the final data set. So that was very much a multi-crystal example. And then a few years later, some of the authors published more details into how they perform the experiments. And so of particular interest here is that they note that they were that in their opinion it was one of the first projects to employ exclusively short wavelengths, which helped lead to considerable improvements in the signal to noise ratio, and also in the crystal lifetime. And for people who were here yesterday afternoon listening to Gwyndaf's talk, it's describing how new beamlines such as VMXM have been optimized to be able to collect at even shorter wavelengths, and they're able to take advantage of new detector technology like the cadmium telluride Eigers in order to be able to better handle short wavelength data. They also had to encounter or be aware of issues with preferential orientation of the crystals, because they were mounted in quartz capillaries. And a final interesting piece about this paper is that, or this example is that they also had to deal with indexing ambiguities, because the space group of the crystals was I23, which means that the unit cell has a fourfold symmetry, but the crystals themselves don't possess the fourfold symmetry. So there's two possible different ways of indexing the patterns, and it's necessary to ensure that all of the patterns are consistently indexed. So in this case, what they did was accumulate a subset of the images that agreed well with each other, and then use them as a reference data set to test all subsequent images against. And yeah, and then in the 90s, Elspeth Garman and others did a lot of work to develop to develop and popularize cryocooling, which then meant that in a lot of cases it was then possible to collect a single data set, or a complete data set from a single crystal. So in many cases where you previously might have needed to collect data from multiple crystals, that was no longer necessary. So given that, why might we still be interested in multi-crystal data collections? So I'll start with a sort of a definition, and I think these are probably quite fuzzy definitions to some extent, and people may have different opinions. But in general, you can, if you're like moving from like a conventional cryo data collection, where over on this side where you might have a single crystal, and you'd be collecting potentially a few thousand images from that single crystal, and you can then sort of view that as a sort of a continuum that as you reduce the number of images that you're able to collect per crystal, you naturally then end up having to collect more, collect combine data from more crystals, and obviously the extreme of that is serial femtosecond crystallography at an X file, where you would typically be only collecting a single image per crystal, and you would need to merge together thousands or tens of thousands of crystals. And I think it's Graham's definition of what is serial is when you've got too many to count. So one reason why you might need to collect multiple crystals is if for whatever reason it's only possible to collect an incomplete data set from a single crystal because of radiation damage or crystal lifetime. So in that case, the solution is to obtain a complete data set by combining data from multiple crystals. So that might be if you've got particularly small crystals that you've been able to grow, or maybe you have specific reasons for wanting to do room temperature data collection, which reduces the crystal lifetime. So in this picture shows is an example with the Dars reciprocal lattice viewer, where showing the reflections, strong reflections found for individual crystals, and each crystal is colored differently, and you can see that by combining like lots and lots of narrow edges, you can at different orientations, you can gradually build up a complete picture of reciprocal space. And another reason for combining data from multiple crystals is to increase signal to noise for weak data. So if under the assumption that you've only got random errors, then the standard error or uncertainty in a measurement is reduced by the square root of n if you repeat the measurement n times. So if you combine data from multiple crystals, you can hopefully increase, or you can reduce the error in your measurements. So one common application might be for enhancing weak anomalous signal for phasing, and I think it was the classic paper is this paper from Wayne Hendrickson's group, where they showed that by combining that, compared to just a single crystal, they add more and more, combine more and more crystals. Some of the anomalous signal metrics like anomalous CC and delta F over sig delta F increase with the number of crystals, and then also if you look through that into the substructure solution, you can see that from just a single or even two crystals, you don't get enough signal to be able to get any successful solutions. But once you start having 3 and then adding more and more, then the solutions get stronger and stronger. For a more recent example is from the Tom V protein that was solved recently using VMXM. So the fascinating thing about this example is that the wild type crystals that they had been sitting in a freezer for like 60 years or 60 or 70 years, I think. But unfortunately, even like collecting optimized data on I24 and VMXM, they weren't able to solve that with molecular replacement, because there's no close homologs, and so they end up having to engineer some methynenes into a wild type clone in order to form a selenomet version. And then from a lot of careful collection of, I think in the end they combined 67 crystals with dials and ZO2 multiplex, and then followed by crank 2, and that gave a good solution and gave a completely unexpected protein fold. So that I think the structure was a fully helical structure rather than the expected beta sheet. Okay, so another reason for wanting to do multiple crystals is for room temperature data collections. So, for example, some classes of molecules, such as viruses, typically suffer on cryocooling. So, room temperature is basically the only option, or there's increasing interest in doing room temperature studies to see if there's any differences between cryo and room temperature structures. But it's important to remember that at room temperature, the radiation damage occurs at an absorbed dose of 1 to 2 orders of magnitude lower than at cryo temperatures. So there's a recent example from Dan Keady's group where they did a large-scale fragment screen at room temperature, and whilst in most cases the things that bound were in a similar conformation, they did find some differences where some of the fragments either bound in a different conformation, or even in a completely different part of the protein. And then another increasingly popular reason for doing multi-crystal data collections is because you want to do time-resolved crystallography. So that can give you potentially give you biological insight into like whatever reactions are going on inside your protein or enzyme. And there's multiple like different sample delivery methods that have been developed for that, and there's many beamlines and are actively developing that area. So one recent example is from the Jasper Van Thor group from data collected on I24, where using a double laser setup, they were able to probe I think 6 different combinations of illumination states and pH, and investigate. I think it's a reversibly switchable fluorescent protein. Okay, so what challenges do we have in multiple crystal data processing? So one one problem is really like the identification of a consensus symmetry. So in a lot of cases, you may know what the symmetry is ahead of your experiment, but in others it may be unknown, and trying to come up with a consensus symmetry from this can be challenging, and as it was described earlier, that can be complicated by the potential by the presence of a indexing ambiguity. But so new algorithms have been developed to help in this case, and they can work quite well in many cases. As Greta described well, you know, there's a number of different approaches to examine non-isomorphism. So ideally, you don't want to include non-isomorphous crystals in your final data set because it may degrade the final quality. One of the earliest examples of this would be unit cell clustering, and it's also useful to look at the distribution of the unit cell parameters to see, for example, whether a time-resolved experiment is progressing as you expect. There's also, you can do pairwise clustering on intensities, or there's also the delta CC half method that Greta nicely described earlier. There's also preferential orientation. So it can be important to be aware of whether it's a problem depending on your crystal symmetry and morphology and experimental setup. You might, if you're collecting, if most of your crystals are in the same orientation, you'll be collecting the same region of reciprocal space again, over and over again, which is not what you need. That can also be relevant for the fixed target data collections, where we're having seeing like systematic like distributions like here can be indicative of chip overloading, because it means that you've got basically getting a second crystal that's sort of preferentially aligning on the side of the apertures. And then of course then there's also a number of metrics that can be useful for looking at radiation damage, but as we mentioned, the best thing is not to kill your crystals. So I would highly recommend always using rad-dose 3D before your experiment to try and set your data collection parameters appropriately. So just finally, just to finish on a couple of slides on what I see as some of the future challenges that we face. So there's been some developments in recent years. For example, this group out of chess to combine the fixed target serial approach with sample rotation. So rather than collecting a sequence of still images at every shot position, they want to collect a rotation data set, which reduces, as Graham alluded to, reduces some of the systematic errors, and it's possible with this to reduce the number of crystals needed for a complete data set, and it also opens the possibility of combining it with time resolved experiments, and then you can each reflection can be assigned a unique time point based on the refined centroid. Obviously, the sheer volume of these sort of data collections provides challenges in terms of like auto processing for facilities. And then another challenge that we're facing is as faster detection and brighter sources are becoming available, such as the Jungfrau 4M or the Pilatus 4, which will have like 1 kilohertz or 2 kilohertz frame rates, which will be which in some cases may require specialist hardware or some serious rethinking of algorithms for how we can just handle that much data rate and volume, both for the facilities and users. And there's also the possibility of event counting detectors, where rather than counting images as traditional types are, that they're counting in individual like photon events, but that they offer exciting possibilities for time-resolved experiment, looking forward into the future. Okay, so then just to finish, then acknowledging there's a lot of people at Diamond and elsewhere that have helped along a lot of the work I've been doing in this area. Okay, thank you for this.
Thank you very much, Richard, for again a very interesting talk. Do we have any quick questions, please, before we break for lunch? Just trying to cast my eyes around the room. Anything from Richard? It's always fascinating to be able to listen to a talk like yours and look back on historic perspectives in the various problems that we still face or reface. In looking through them, did you come across any useful techniques or applications that might be helpfully reintroduced? Is everything entirely progress?
Yeah, I mean, I think we've got a lot of advances in hardware and software and algorithms that that can help mitigate some of those issues, but we have face a lot of new problems in terms of the scale of the data we have to look at. Although that's a problem they were complaining about, like back in like the John Kendra in 1960 myoglobin paper. They were complaining about the size of the amount of data that they were collecting, because now they were collecting high resolution. They needed to use this latest and greatest like EDSAC Mark II computer or something.
Anyone? No? I think in that case, it looks like people are anxious to get to their lunch. So if you have any further questions, perhaps take them up with Richard later.
Right. Okay.
Brilliant. Quickly, just to thank all the speakers in this morning's session.